Sir, shall we start? You can start. Sir, shall we start? Sir, shall we start? You can start. 1 second, 1 second.
Sir Vasant Kumar, sir, there is a background noise. Sir Vasant Kumar, sir.
Yes, sir. Yes, sir. I'm.
We are live. There's a background noise around you, sir.
Yes, please. Sir, is it visible, sir? The screen is visible?
Screen is visible, sir.
Okay. Can I start, sir?
Yes, sir, please. Sir, please avoid background noise around you, sir.
Yes, sir. Very good morning to all of you. Welcome to you all for the third day of the Virtual International Conference, ICA MASM 2020, jointly organized by the Departments of Mechanical Engineering, Aeronautical Engineering, Automobile Engineering, IEM, and MBA at the Sagar College of Engineering. So we now have a plenary session 4. Today we have with us one of the distinguished alumni of the Sagar College of Engineering, Dr. Srinivas Hanasoge. And it's my privilege and joy to give a very brief introduction to Dr. Srinivas Hanasoge. Qualifications wise, he is our alumni from the class of 2011 who graduated from the Department of Mechanical Engineering. And later on, he did his Master of Science in Mechanical Engineering at the Rutgers University at New Jersey. At Rutgers, he worked on formation of multiple micro vortices in regions of ion concentration and polarization as his master's thesis, and also worked on size-based microparticle separation. And later on, he pursued his doctoral work at Georgia Institute of Technology, Atlanta. And his thesis work was on the design and fabrication of artificial magnetic cilia for lab-on-a-chip applications and 3D multiphysics modeling of composite thin film membranes. And it's my privilege and joy to say that he also continued his research as a postdoctoral researcher at the Georgia Institute of Technology, where he developed a high throughput microfluidic pattern for cell mechanoporation for the delivery of macro molecules into cells, design and fabricated silicon glass-based microfluidics devices for high pressure microfluidics. He has a number of research papers to his credit in terms of various popular reputed journals like Lab on a Chip, couple of publications, Microfluidics and Nanofluidics journals, Nature, Micro Nano, Soft Matter, International Journal of Performance Analysis in Sport, and a good number of publications journal publications to his credit. And he also has the teaching experience at Georgia Tech as an instructor, both in lab and theory. And most important is the award that has been displayed here as an outstanding paper of microsystems and nanoengineering at Springer Publications Nature 2020, spotlight article in Nature Physics, and elected member of the Sigma Scientific Research Honor Society, and third place in Electrochemical Society Student Chapter and annual poster competition. So it is my privilege and joy to introduce Dr. Srinivas Hanasoge, and his skills are on microfabrications, microscopy, multiphysics and CAD modeling, and his interests are in sensors, wearables, lab-on-chip devices, microfluidics, MEMS, microfabrication, design of experiments. He is going to deliver his plenary session on the same topic. So on behalf of the organizing committee of this international conference, ICA MASM 2020, it is my privilege and joy to heartily welcome Dr. Srinivas Hanasoge to our conference. Welcome, Dr. Srinivas. And I now request you to deliver the plenary session address on the topic recent trends in microfluidics, MEMS, and microfabrications, live from San Francisco, USA. Over to Dr. Srinivas.
Thank you so much for the very kind introduction, Vasant sir, and the very nice bouquet as well. Much appreciated. So I'm going to take over now and I'm going to share my screen. Can you see my screen?
Yeah, it's going this case.
So I'll be talking about recent trends in microfluidics, MEMS, and microfabrication. What I'll be covering primarily is the work that I did as part of my master's, PhD, and my postdoctoral work. But before I begin, I'd like to thank the organizing committee, especially Dr. CPS Prakash sir and Dr. Vasant Kumar sir for inviting me. I'm very honored to be here today. I feel at home at Dayanand Sagar, so I'm very happy to be here today. So let's get into it. Briefly, okay. A brief background about me. Give me 1 second. Okay. A brief background. I graduated from Dayanand Sagar in mechanical engineering in 2011. And then I did my master's at Rutgers, and then I went on to do my PhD and my postdoc at Georgia Tech. I'm currently working at Verily Life Sciences, which is part of Google X as a hardware engineer. And briefly, here is what I'm doing right now. What we're trying to do is develop a smart contact lens for multiple different applications, some of which could be for continuous monitoring of blood glucose. Some of it could be active accommodation of vision based on where you're trying to look. So these are all the different projects that we were working at Verily. And we have started a new company called 2020 Therapeutics, which is a spin-off of Verily. And I'm currently working as a hardware engineer for that company. So that's briefly what I do right now. And what we'll be talking about today is primarily my work that I did for my master's, my PhD, and my postdoc. So what is microfluidics? Microfluidics basically refers to manipulation, control, and basically transport of fluid dynamics at a micro scale. And when you say micro scale, it could either be length scales or volume scales. So it's essentially talking about a very small amount or quantity of fluid. How we can manipulate this, how we can go about doing any kind of analysis on such scales of fluid is what we call microfluidics. Why it is important is something that I'm going to try and tell you today, and why we should be focusing more, or why this is relevant, especially in today's age of coronavirus, is something that I'm going to try and tell you. So there are multiple aspects to microfluidics, and the most important one is obviously that we're going to be working with small volumes of fluid, and the micro domain effects here are going to be more significant. And what I mean by that is, as the volume of fluid that we are considering is reduced, the importance of surface forces, the importance of surface related effects are going to be more prominent, because the surface to volume ratio is going to be much larger when the volume of the fluid is much smaller. Because the surface area of a sphere is going to be proportional to r square, and the volume is going to be proportional to r cube. So smaller the radius, the more important the surface area effects. Because of this, and because of the presence of viscosity, which is going to be significantly more significant than the fluid inertia, the domain of microfluidics is kind of separate from the rest of fluid dynamics. It's not going to be any kind of turbulent flow. It's not going to be any kind of inertially driven flow. And I'm going to tell you a little bit about what that means and why this is significantly different from the traditional fluid dynamics that we tend to study. So what are the primary applications? The biggest use for microfluidics is in lab-on-a-chip technology. The idea here being all of the processing and all of the laboratory work that you would potentially get done. So let's say you give a sample of blood or urine or any kind of fluid to a lab. You basically need to give a large volume of fluid, and you need a full-scale laboratory to perform any kind of analysis on the sample that you provide. Once they perform the analysis, they'll give you the result. Now, the idea is basically to condense all of this into something that is on a chip form factor. So this is going to be a point of care diagnostics where you can actually take the device to the point where you want the diagnostics to be happening. So you give it out to all the patients that you need. They should be in a position to be trained and use these devices themselves. And these are ideally going to be disposable devices or something to the extent of that, where the patient is able to self-diagnose, the patient is able to take these tests at home or at the place wherever is convenient, and they'll be able to get some results which are clinically relevant. So that is the primary idea behind lab on a chip. So you take a lab, you condense it onto a chip. And this is possible with microfluidic technology because we are currently in a position where we can take a very small volume of fluid and perform different kinds of reaction, different assays, and tell what's the constituents of the fluid. If there is any kind of disease, we are able to tell that. A typical example of an already commercial product is the standard blood glucose monitoring device. You can take the test. It's essentially a prick test. You prick your finger with and get a drop of blood, and it's able to tell you what or how much the concentration of blood glucose in that particular sample is. The same thing with the pregnancy test as well. You are able to tell if you're pregnant or not, this diagnostic can happen pretty much anywhere. So the primary characteristic of a lab on a chip device handling a very small volume of fluid is essentially that the viscous forces here or the viscosity of the fluid is particularly dominant. And what I mean by that is in comparison to inertial forces, the viscous effects are going to be dominant. And this is particularly important because all of the flow here is going to be considered as low Reynolds number, creeping flow. And what I want to do today is talk about two different instances of how people tend to study microfluidics. One is from a lab on a chip point of view. I'll give you an example of how you can combine multiple different ideas to have basically a CAR-T therapy, which is a cell engineering therapy for the cure of cancer. I'll walk you through how the device design is done for that, and basically what are all the different components for making such a lab on a chip device. And the second point is, and the second point of view for studying microfluidics is understanding biomimetics and using biomimetical systems to basically emulate and to make your engineered systems a little bit better. So I'll talk about both of these. We'll start with the microfluidics for a lab on a chip technology. So what is cell engineering? Cell engineering is essentially you take the cells of a human being, you engineer those cells, and you give it back to the human being, and you expect these cells to work in the human being's favor. In particular, what I'm going to talk about today is called CAR-T therapy. CAR-T basically stands for chimeric antigen receptor therapy. And T here stands for T cells, which are your body's immune cells. So the idea here being, as shown in the diagram here, the idea here being you take a sample of the blood of the human being who wants to be treated for cancer, assuming that he has cancer. You isolate the T cells. You isolate the T cells and you program these particular cells to have a certain kind of receptor on its surface. Essentially what you're doing here is you're going to program the cell to specifically attack the cancer cells. So that is the idea behind programming it. I'll walk you, I have a couple of slides on what it means to program a cell. So essentially we'll be changing these cells and giving them the ability to attack specifically the target, which is the cancer cells. Once you do that, you basically multiply them, which is you do a cell culture and you multiply these cells. And once you have enumerated them and once you have amplified the amount of cells you have, you basically inject it back into the human being. And what that does is now these cells, which are specifically capable of attacking the cancer cells, will essentially go to the cancer, and you have a much higher chance of surviving the cancer. And this is particularly useful as compared to the traditional chemotherapy and the radiotherapy treatments, primarily because there are no side effects. You're using your own body's immune cells. And this is not a brute force method as the traditional chemotherapy or the radiotherapy. Both of which methods either use electrochemical radiation, electromechanical radiation, or they use chemicals, which basically are brute force methods for killing the cells, which would also be killing your healthy cells. So the advantage of this CAR-T therapy is primarily that you can use your own cells, reprogram them, and have a better chance of overcoming cancer. So how we are able to do this using lab-on-a-chip technology is what I'll be talking next. So the first thing that is required to be able to do this is isolate T cells. T cells are basically lymphocytes that are white blood cells in the blood. The first thing that needs to be done for you to be able to do this technique is you have to be able to isolate these cells from the rest of the blood. If you take whole blood, it has different constituents to it and different kinds of cell types in it. The picture here basically shows you everything that's in it. We have blood platelets, you have red blood cells, you have white blood cells, you have lymphocytes, you have a whole variety of different things. One thing that stands out is all of these cells have different properties. One of them, which is of primary importance here, is these cells have different sizes. So you can use the fact that these cells come in a variety of different sizes to separate them. So the technique that I'm first going to talk about is essentially a size-based separation of cells that can be done to basically isolate the T cells in the whole blood. So to give you a sense for what the sizes here are, the T cells are on the order of about 25 microns in size. 25 microns is about 1/4 or 1/5 the size of the width of the human hair. And the red blood cells are about 3 times smaller than that. So there is a 3 times size difference between the T cells and the red blood cells. Red blood cells are the primary constituent of the blood. So the key here is to basically separate out these different species based on their size. There are multiple different techniques that are available for doing this. And what I'll be talking about today is specifically called deterministic lateral displacement, where you're basically running the device through a microfluidic device, and you see that different particles of different size migrate to different positions. Here we see a video which basically demonstrates the same concept. We have a main fluidic channel through which multiple constituents are flowing, and you have an array of obstacles inside of the channel. These particles basically, or these cells, interact with these obstacles, and because of their size difference, the way they interact with these obstacles is going to be slightly different. Because of which, over the length of the array, over the length of over the length of the entire periodic set of arrays, you will see the particles of different size and shape migrate to different positions. So if at the outlet of the channel, you will basically see that particles of different size have migrated to a different lateral position, which enables them to be sorted out. So that is the idea behind deterministic lateral displacement. So again, to reiterate my point, on the top here is an animation of what's happening. So we see an obstacle. The obstacle is represented by the circle here, shown in the center. And we have 2 particles. We're going to look at 2 particles. One is a large particle and one is a small particle. We'll first look at a large particle and see how that large particle interacts with this obstacle, and then look at a small particle and see how that interacts with the obstacle. So a large particle starts at the same position as the small particle, and as it transitions, it interacts with the obstacle, and it ends up in a certain lateral position post the obstacle. Secondly, if a small particle starts off at the same lateral position, it interacts much differently with the obstacle, and it results in it being at a different lateral position. So if you can see my mouse, post the obstacle, you'll see that the large particle ends up in a different position and a small particle ends up in a different position. And this is a deterministic behavior, and this is going to be determined by how big the particles are and how big the post is or how big the obstacle is. So essentially, you can use this technique and you can have an array of these obstacles, and making the particles go through these array, what you can essentially get is over a certain number of arrays, you can have particles of different size migrate to different lateral positions. On the bottom is an example of the same thing and what happens over a number of arrays. You see that a large particle basically follows the path shown in green here, and a small particle basically follows the direction in which the fluid is flowing. So essentially, at the end of the channel, these have migrated far away from each other, and you can basically separate them out or collect them in different containers, and the species of interest is essentially separated. And how is one of these devices made? So here is briefly the fabrication process for it. It's pretty standardized fabrication process using simple things like glass slides and simple photoresists and simple lab equipment. There is nothing too sophisticated here. Primarily what's happening here is we use something known as a cast molding process, where we make these micron-sized obstacles. We make a mask, and we cure a silicone elastomer on top of the mask, and we get these obstacles, which are then bonded onto a glass slide. And what you essentially get is a device that has pores. So you can think of this as essentially a glass slide with small pin fins or small pillars on top of it. That's essentially what this looks like. And then you're going to be flowing fluid on top of it using a very small micro channel. And like I said, when the particles interact, you're going to see them interact with a certain periodicity. And in my work here, what we have shown is we are able to do this with an electrically driven flow. So the fluid flow here is driven electrically. And we see that particles of a certain size interact in a deterministic way. So they have the same periodicity. So all of the particles that you just saw cross basically move 2 to the right and 1 above, which means that they have a set periodicity and a set angle at which they migrate. This is going to be different for particles of different size. So in this case, the larger particles are going to be migrating at a certain angle, and the smaller particles are going to be migrating at a different angle. So this essentially leads to separation of particles over a certain distance across the array. Okay, so what's next? So let's say we have isolated the T cells. Now we need to engineer these cells, or we need to make sure that these cells can specifically attack the target species. In this case, let's say a cancer cell. So the industry standard for how some of these cells are engineered is called electroporation. The idea here being you use electricity to create physical pores on the cell membrane, and in the vicinity of the cell are going to be the species that you want the cell to be absorbing. So you can think of it as essentially injecting certain DNA or RNA material into the cell. And when you insert the certain DNA or RNA material into the cell is going to express the receptors that you want on the surface. And now if you put the right DNA or RNA inside the cell, you're going to have it express the right thing on the surface, which is going to make it specifically attack the cancer cells. That is primarily the idea. So the current standard which is used, which is the electroporation technique, basically applies an electric field across the cell. Because of the strong electric field, the cell membrane is going to create, there is going to be pores created on the cell membrane. When the pores are created and the cell is basically suspended in some kind of a fluid, which has the mRNA or the RNA or the DNA material in its vicinity, these are going to be absorbed into the cell body. Once these are absorbed, the cell is able to express the right receptors, and it's going to be specific in its target. So that's basically the idea for how a cell is programmed. And what we tried to do was use a technique called mechanoporation. So instead of using an electric field, what we wanted to use was mechanical forces to deform the surface of the cell and create pores on the surface by simply applying mechanical forces on the surface of the cell. So here is a picture of of the working principle. The fluid flow is going from the left to the right, and we have a microchannel through which the cells are being flown. The cell, the leftmost point here, is a cell in its natural state. It is forced to go through a constriction. Essentially, it is being squeezed through a small constriction. And when it does get squeezed, there are going to be pores opened up on the surface of the cell, and all of the material that is outside of the cell. So if you have some genetic material outside of the cell, these are going to be absorbed into the cell interiors. And you do this periodically. You do this over a period of multiple different constrictions until a point where you know enough of the genetic material is inserted into the cell. On the bottom here is an image of fluorescent particles. So the cell is about 25 microns in diameter. And we injected the cell with some fluorescent particles, which were much smaller, on the order of about 500 nanometers. And you see that the fluorescent particles have actually gone and they have been injected into the cell surface. So instead of fluorescent particles, if you did the same thing with genetic material, you would essentially have reprogrammed the cell to do what you wanted to do. So that's essentially the idea behind cell mechanoporation. And combined with cell sorting and cell separation methods and cell mechanoporation and techniques like this, we are able to create lab-on-a-chip devices which are high throughput, meaning that you can process a bunch of cells, or you can process millions of cells on a small disposable device, and there is no sophisticated equipment or machinery needed to run a process like this. So that is primarily the idea behind lab-on-a-chip technology. And here is an example of what the fabrication process for a device like this looks like. The fabrication wise, it's pretty similar to what you saw before. Essentially, instead of making obstacles, what we have made here is constrictions. And you basically make the cell flow through these constrictions. And on the top right is a confocal image showing you the depth data of what the microchannel looks like. And in the bottom are some simulation results for how much or how the cell is getting squeezed as it goes under each of these constrictions. Okay, so next. The next thing that I want to talk about is microfluidics from a biomimetic and a biological physics point of view. Why it is important, why it is relevant. So first of all, like I said, microfluidics is an inertia-less environment, meaning that the viscosity is going to be the primary driving force. There is not going to be any fluid inertia. Further, the fluid mixing is going to be dominated by diffusion. It is not, again, going to be convectively mixed. It is going to be primarily diffusion mixed. And what I mean by that is demonstrated pretty well in this video. Many of you might have seen something like this before. So what's happening here is we have two cylinders between which there is a very viscous fluid. And the operator here is primarily adding three dyes of different colors. One is red, green, and blue. There are three dyes of different colors that are added into the space between the two cylinders. There is an inner cylinder and an outer cylinder. Again, like I said, the fluid here is very viscous. And what he does is he's going to rotate the inner cylinder with respect to the outer cylinder. So what's happening here is you can think of it as Couette flow between two plates, but essentially it's happening between two cylinders. When you spin it, it appears like all of the fluids are being mixed. So he does a few different rotations, and it appears that all of the three different dyes that he put in there are mixed. After a certain number of rotations, he is going to reverse the number of rotations to the exact same point. So if he did 3 rotations clockwise, he comes back 3 rotations anti-clockwise. And what you see, surprisingly, is the fluid basically gets unmixed. So what's happening here, so all the 3 colors are now separated in spite of them having appeared to have mixed. So what is happening here is, although the fluids were moved, the fluid was basically folded on top of each other. It was not merely mixed. So this is the reversibility of a viscosity-driven flow. And this is primarily the environment in which microorganisms or most of the fluid handling in a microfluidic device is going to work at. So since this is macro scale, and hence we had to increase the fluid viscosity to get the same effect. But you can think of a small organism living in an environment that has this high viscosity, and where the fluid flow is based, the fluid flow is basically reversible. And here is another example demonstrating the same concept. What we have here is a reciprocating swimmer. You can think of it as a fish which is flapping its tail back and forth. The video on the left is basically going to show you how this fish or the reciprocating swimmer is capable of moving forward by moving a symmetric reciprocating motion. So it's able to propel itself forward. Essentially what's happening here is it is pushing the water back and using the fluid inertia to propel itself forward. And this is a high Reynolds number flow. I wouldn't call it turbulent, but it's not Stokes flow, it's not creeping flow. On the right here is the same reciprocating swimmer, but now suspended in a fluid that is much more viscous. Now, because of the increased viscosity, what's happening here, although the swimmer is doing the same reciprocating motion, what the motion is doing is primarily just moving the fluid back and forth. Because the fluid is just being moved back and forth, and there is no fluid inertia that the swimmer can make use of, it is incapable of going forward. It is incapable of pushing itself forward. So this is primarily the difference between macro scale swimming and micro scale swimming. And this is also the reason why people tend to drown in quicksand. Quicksand is a regime which belongs to the low Reynolds number. So essentially what's happening there is you cannot swim like you would in a swimming pool to get out of quicksand, because a mere reciprocating motion is not going to be propelling yourself because it is an inertia-less environment. So how does nature solve this issue? We know that there are multiple things in nature which operate in this microfluidic environment, and they are known to be excellent swimmers. For example, sperm cells are supposed to be excellent swimmers. We know that. But they are operating in this regime, but how are they able to propel themselves is an interesting question. Here are a couple of other videos of microscopic organisms which are using which are using organelles such as cilia and flagella to basically create fluid flow. The video on the right bottom here is a starfish larvae, which again is a very small organism on the order of about a few hundred microns, that is a couple of hair strands. And what's happening here is this organism is able to create these interesting flow patterns around its in its vicinity, and it's basically using this flow for eating, feeding, movement, and all of these different activities. So how are these microorganisms, how are they capable of creating such fluid transport in spite of being in an inertia-less environment? The answer is in creating an asymmetric motion. So what's happening here, the image on the left is showing you how essentially a fish is going to swim, or any large-scale animal. So what's happening here is a fish basically does a reciprocating motion, and it's a symmetric motion, and it's a two-dimensional motion, and it's able to propel itself forward by using fluid inertia. However, a sperm cell or a small organism which is capable of moving is going to do the same by using something a little more complicated than just a to and fro motion. What it actually does is called, it's a helical motion, where there is an inherent asymmetry to the motion. So it's essentially like a corkscrew, as shown in the picture below. It's not a mere reciprocating motion. It's a three-dimensional motion in which the tail is basically going in a helical fashion. So because of this, microorganisms and most of the microorganelles that the organisms use are capable of creating fluid transport in their vicinity. So natural cilia is the most commonly used organelle by different organisms and even in human beings. If you look at the inner linings of the nasal tract or the stomach linings, these are filled with hair-like structures called cilia. And these filament or these organelles basically are hair-like organelles, and they create fluid transport in this microfluidic environment. And they are capable of doing this by creating an asymmetric motion in their beating. And what I worked on for my PhD was artificial analogs of these cilia. So we made magnetically actuated artificial cilia, which I'll be talking about in the next few slides. So here is briefly the fabrication process, but I think I can skip over that and tell you a little bit about how it works. So think of this as a bunch of hair-like filaments. These filaments are going to be made of a magnetic material, which means if you put a magnetic field around it, it is going to get magnetized and it is going to try and follow the magnetic field. So the drawing here basically shows you a bunch of these hair-like features, and then you put a magnet in its vicinity and you oscillate the magnetic field. You either rotate the magnet or you move the magnet around. These filaments have high deformation and they're capable of moving significantly. Here is a video of what happens. So these are large amplitude oscillations of these filaments as a magnetic field is being oscillated or varied in its vicinity. So these are all magnetic filaments which get magnetized in the presence of an external field. So that's nice. We have some filaments which can move around, but what we really need to create fluid transport is for them to have an asymmetric motion, something similar to what the sperm cells or some of the other organisms use. And what we did was we tried to image the trajectory or the motion of these individual filaments. And what we found was indeed they do have a time asymmetric motion, meaning they follow a certain trajectory in the forward stroke, and in the recovery stroke, they follow a completely different trajectory. So this difference in trajectory is not the same as a reciprocating motion, and it therefore provides the asymmetry required to create the fluid transport in the microfluidic environment. So that was essentially the video showing. And the path here shown in white is the forward stroke, and the path shown in yellow is the recovery stroke. Since these two lines don't coincide, there is going to be a net fluid displacement. We did some simulations to compare this. So we are at a point where we understand the kinematics and the dynamics of these filaments. So these are some COMSOL simulations which show how there is an inherent asymmetry for a filament that is actuated magnetically like this. And because of this asymmetry, we are able to create some fluid flow. And here is a video of what happens if you have, so essentially what you see here is a microfluidic channel in which there are some fluorescent particles which make it easy for visualization. And there is an array of these filaments. So these arrays are, so the cilia here are facing one direction, the opposite direction, go back in the same direction, and this faces this direction. So when you actuate the cilia, you should expect to see some fluid motion. And that is indeed what we see when we actuate the cilia. Because of the asymmetric motion, it is capable of producing a net fluid transport or a net fluid pumping in the microfluidic environment. Now, why is this important? It is important because most of the fluid handling that we do for a lab-on-a-chip device, let's say you want to perform a reaction, or let's say you want to capture bacteria, or for some application, you want to create localized transport or localized mixing of fluids. It is not going to be easy unless you use some kind of an active mechanism similar to what I showed here. So this helps create more microfluidic transport, which in turn can enhance your reaction and in turn can be beneficial for performing a bunch of different operations. Further, naturally occurring cilia don't all beat together. So these hair-like structures on the human body or on different animals basically beat in a metachronal fashion. And what that means is they beat one after the other. The millipede here is basically walking with a similar metachronal motion where you see that all of the legs are being moved one after the other, and they're not going to be moving simultaneously. And the same thing is shown in the cilia here on the right, where the cilia are all beating one after the other. And this can often be seen in football stadiums or cricket stadiums where they do a Mexican wave where it looks like a traveling wave on the in the audience. So this is how most naturally occurring cilia and organelles function. It is important because there are known to be specific advantages of these organelles behaving this particular way. And what we were able to show is in our artificial cilia, we are capable of producing a similar metachronal motion by primarily varying different properties of the filaments. So these filaments here are basically increasing in length. Because they're increasing in length, you see that there is a phase difference in their operation, and they are actuated one after the other in a sequential manner, similar to what you see in naturally occurring cilia. So this system basically gives you a platform for exploring why naturally occurring cilia do such metachronal motion. In addition, we also explored a couple of other ideas where here what you see is an array of cilia which are all being actuated simultaneously. And now we electrostatically hold some of the cilia down, so some of them are held down while the others continue to oscillate. So with this technique, we are capable of selectively actuating certain filaments if you have an array, and you get more control over what can be actuated and what cannot. So that could also prove useful. And speaking of an application, some of these artificial cilia could perhaps be used for detecting bacterial cells. So bacterial cells which are on the order of about 1 micron, which is 1/100th the size of a human hair. Let's say you want to capture it and you want to be able to detect it for multiple different applications. So in this case, what we have shown is we captured Salmonella bacteria, which is a bacteria that causes stomach and gut issues. We were able to capture this bacteria on the surface of these filaments simply because these filaments were capable of creating microfluidic mixing and microfluidic transport on the order or the scale of the size of the bacteria, with which we were able to functionalize the surface with the correct antibody, and we were able to capture these cells on the surface of the cilia. So that's what this is. Other examples of lab-on-a-chip devices. So here are a bunch of other things. I hope I've conveyed the point of or the importance of microfluidics, how it is relevant, especially in these days. There are a bunch of papers on lab-on-a-chip devices that people are trying to develop for rapid COVID detection. And here are a bunch of other examples of the different other interesting things that people have tried to do. The first one here on the top left is a PCR device that was made for real-time PCR detection for Ebola virus. So with this device, you will be able to tell if a person has Ebola or no. And it's basically a handheld device which does the DNA amplification, and it's able to tell you quantitatively what the results are. On the right here is a new technique called lab on a PCB, similar to lab on a chip, where you have fluidics and electronics integrated onto the same chip. So you can perform a bunch of electrochemical reactions. You can do lots of things with this. And what they particularly demonstrated was detection of virus. So the surface, there is an active surface here which primarily acts as a capture point for the virus. And once the virus is captured, you are able to detect it using some of the techniques they have developed. On the bottom here is a very interesting concept called a paper centrifuge or paper fuge. The idea here being the primary idea here is to replace a benchtop centrifuge in resource limited settings. So what they try and use is basically a paper toy, which is connected with 2 strings, which can basically spin at very high RPMs. So you use this toy, which is maybe 10 or 20 rupees, and you can create a microfluidic channel on, or you can create a microfluidic channel on these discs and basically separate the blood plasma and the other blood constituents just based on the centrifugal forces. And this was also used, I believe, for the detection of malaria. So that's a really frugal way of detecting something as complicated as malaria, and that I'm sure is going to be very successful for multiple different applications in resource limited settings. So in conclusion, In conclusion, I hope to have made the importance of microfluidics in point-of-care diagnostics and lab-on-a-chip devices clear. The estimation for point-of-care devices is it should have a market share of about $4.5 billion by 2023. Initially, it started with the semiconductor microfabrication technology that was being used for making these devices. However, the good news is microfluidics now shifts from clean rooms to maker spaces. And this is something that I want to highlight. And I believe Dr. Omkar as well said the same thing in his talk. He said the most important thing is for the university labs, the machine shops, and all of these places to be buzzing. So I believe that you don't need a lot of resources to try something like this. And that definitely should be possible in resources. We don't need a sophisticated clean room to make these devices. These can be done on a benchtop. And it seems like the technology is moving towards organ on a chip or a body on a chip, or techniques like this where drug discovery could be crucial. You don't have to use an entire human as a patient for telling if a particular drug is going to work or not. You could essentially do that by using some of the cells. So that's essentially what I wanted to talk about today. With that, I'd like to thank you all for joining me and being patient listeners. I'd be happy to entertain any questions.
The session is open for question and answers. Participants, welcome to ask questions, or you can just type it in the chat box also.
You can unmute your mic and speak. Alternatively, you can also post a question in the chat box.
I have a question, Dr. Srinivas. I was able to see a very wide and important application, medical sciences, in about is microfluidics. But what are the other areas of applications that it could be used very successfully and potentially?
Right. So one primary other application of microfluidics is in printing technology. So people are doing picoliters of droplet dispensing for printing, and this is already being widespread. And you can also argue that inkjet printing is a form of microfluidic technology. So there is a lot of applications in printing. But primarily, microfluidics is the primary interest is in the biomedical and the medical research communities. And there is a whole bunch of other applications. So people do droplet microfluidics for a huge amount of different applications. Yeah.
Can you please throw some light on your present work at Google, Verily Google Life Sciences?
Sure. So what I am currently working on is develop a contact lens, which is capable of accommodating vision. And what that means is the glasses that people, the most commonly available glasses are bifocals, which means that if you look through the top, you're going to be able to see a further away distance. And if you look through the lens region, you're going to be able to focus on whatever is close. And the idea behind the smart contact lens here is it automatically detects what you're trying to look at, and it focuses your vision based on that. So it's essentially going to take the it's essentially going to aid in vision for people who are trying to, or for people who are presbyopic. So I work on developing the microfabrication process for making these devices, and the important project that I'm working on is an offset of this main project. which is also based on the lens, and we are trying to develop a microfabrication process for that. So you can think of it as basically integrating electronics with all the different components onto a form factor, which needs to be flexible, which needs to be oxygen permeable, which needs to be able to fit on the eye and be comfortable on the eye and be medical grade and all that.
Srinivas, there is a question on the chat box.
Okay. How do you differentiate between what has been done with Verily and Neuralink? So I think these are very two different things. So what Neuralink, I believe, is working on, again, I don't work there, so I don't know, is they're trying to do a deep brain implant for recording and controlling what happens in the brain. What Verily is working on, and particularly my product, is related to ophthalmology and what happens on the eye. And Verily works on a bunch of other projects, many of which are public. And I don't see Verily and Neuralink being competitors here. They are targeting very different aspects of the market. Yeah. You're welcome.
Any other questions from the outside? There is one more question in chat.
Yeah, so I have another question here by Purab. He's asking if the electronics on the contact lens won't affect the vision. So if you look at the eye, primarily what you have is the pupil. Pupil is the region through which the light is able to penetrate through and hit the back of the eye for you to be able to detect it. Most of the electronics we have are going to be around the pupil. So it's going to be in the region where you have the iris, and which is opaque to begin with. So this would not be interfering with your vision. You're welcome. For old and elderly people. Well, contact lens is the other question I have here by Akash is is the contact lens feasible for old or elderly people? I think it depends. There is no one straight answer to this. There is no one straight answer to this, and it really depends on. I know a lot of old people who prefer wearing contact lenses. That's not to say that everybody is going to prefer it. I'm sure there is going to be a big resistance in people wearing. I, for example, would not want to wear contact lens. So, so there is a market for it. There are people who prefer a contact lens. There are people who don't. So.
Hello, Dr. Srinivas.
Hello, sir.
This is Professor Prabhakar Kupali. Remember me?
Of course, sir.
Oh, very good, very good. You see, on the cancer treatment, the CAR-T treatment which you are talking about, why people are still going through a very strenuous process of chemotherapy and radiation and all that. They are being harassed, universally with this treatment. And people, it's not just the treatment alone. It is the people who are subjected to all sorts of problems, depression later on, and resulting in death also sometimes. Why it is like that, and why not your treatment? Why is there a solution for this?
Well, in the near future, I hope there will soon be a solution. But the current state of the field is such that there is still research to make sure, and there are actually clinical trials happening right now so that an engineered therapy like this can work. And the result for the first patient who completely recovered from cancer was as recent as 2017 or 2018, if I remember.
I see.
So the first patient who actually got treated from an electroporation therapy was actually in 2017 or 2018. The other drawback of the electroporation therapy was the fact that it's a very expensive technique. It's not cheap. With mechanoporation, and there are other techniques similar to this, the hope is basically the cost of such a service is going to be significantly lower. So everybody could afford it. So currently, such therapies do exist, but they are very expensive. One of the primary motives for the lab-on-a-chip community is to make these therapies available at a much lower cost.
Thank you. Thank you very much.
Sure, sir.
Yeah.
Dr. Harish, can we end if there are no questions?
I'd briefly like to thank everybody, the organizing committee, for inviting me, specifically Dr. Prakash, Dr. Vasant Kumar, and Dr. Kupalli Prabhakar. All three of you have been my mentors, and I highly appreciate this gesture.
Two more questions in the chat box.
And one more question in the chat box, no?
Oh, I don't see it.
No.
So does the use of contact lenses feasible for old and elder people?
I did answer that. So it is feasible from older people get prescribed contact lenses all the time. So it really is up to the user if they want to use it or no. Some people prefer using contact lenses, some people don't. For the same reason, some people prefer using contact lenses, some people prefer using glasses. So it really is up to the user. Thank you, sir.
I think no more questions, so we can proceed.
Hello.
Yes, there was a
Yes,
question.
sir. Can I ask the question?
Yes.
Please, Tejas, go ahead. Tejas, go ahead.
Yes, sir. Yeah, you are talking about lab on a chip.
Mm-hmm.
So basically, what I understood was, so any kind of blood or fluid is placed on the chip. Am I right?
Correct.
Yeah. So when we place the blood or any kind of other fluid on the chip, so how does the chip detect? Does it have a sensor, or does it have a reactive material which reacts to it, or anything else like that?
Exactly. So there are multiple things that you want to detect, right? For example, let's say you want to detect blood glucose, or let's say you want to just tell if the sample is there. Let's say you put a drop of blood and you want the device to know if you have the drop of blood in there. The first thing that you would do is probably do a conductivity test to see if the sample that you have actually put in there is blood. So that's one of the primary things that the device will do. So there is a bunch of electronics that go along with the fluidics to basically establish multiple of these reactions. And if you're measuring blood glucose, for example, there's going to be some electrochemical reaction which directly quantifies the amount of blood glucose. And that is going to be or displayed to you in some form.
So there is no need for any reactive material to be placed on the chip?
There is. So that's why the key here is many of these devices are single use. So let's say you're talking about a blood glucose monitoring system, right? Essentially what's happening there is you use a disposable tip and you connect that to a handheld electronic device. I don't know if you have used one of these, but that's essentially how it works. So it's the strip is a one-time use device. What it essentially does is you put the drop of blood in one end of the device and you stick the device in the electronics that you have. And what that is capable of telling you is the amount of blood glucose you have. And the strip that you used is basically a one-time use. So there are some particular antibodies or some particular chemicals on there which specifically tell you how much blood sugar is there in that particular sample.
So it's basically the same concept, but it is in a very micro, the size is very less.
I'm going to say it is the exact same concept, but not for blood glucose. There are other things that we want to know. What if we can get a small sample of blood and tell if you have cancer? Or what if you can get a small sample of blood and tell a whole bunch of variety of different things. So instead of you having to go, let's say, to the clinic and give 10, 15 ml of blood and then expect the results 2 to 3 days later, instead of that, you could have a test at home kit where you basically prick a small amount of blood and you get your diagnostics in 2 minutes on your cell phone.
So, sir, does this the lab on a chip device have the ability to tell, detect many ailments or diseases or many aspects regarding the blood or any other fluid in one test, or
That's
is it
possible.
like you have to use separate, separate devices for one use? Is it like that? Is it able to do multiple things?
Well, ideally, right? I mean, this is still technology, it's still in development. If there is one solution for all questions, then that would be the preference. But I don't think there is anything close to that out in the market yet. So there are individual devices that people use for different applications. You can think of it basically as an integrator of all these different ideas to give you one device that gives you a result for everything. There was this company called Theranos, which turned out to be a big flop. You can look it up. It was a big story.
Sir, I've
Yeah.
noticed that. I've noticed that. Thank you. So
They
this is
tried to
basically
do
one
all-
use for one case study.
I'm sorry, what's that?
So this is one use device for a single aspect to know. We have to like use only once for a single, like you told, for example, blood glucose. So it only
Again,
detects blood glucose, or can it detect multiple? That was my question. So
the
is
blood glucose monitor is designed to detect only the blood glucose.
only the blood glucose. Okay, sir.
Design something that detects both the blood glucose and something else, polysaccharides or something in the blood.
Okay.
You have a new device which can do two things. So when we talk about lab on a chip, it's not a particular device, it's technology. So what we're talking about is total analysis systems as a whole. So you want to get a sample of blood and see if you can do something similar to what you could do in a laboratory. Could you do all of it or could you do a part of it? These are all good questions, and these are all questions that researchers are trying to answer. There is no answer to this.
Okay, so thank you.
Shall
Kupalli
we?
again, one more question.
Yes, sir.
Dr. Srinivas, a person who has got a cataract in the eyes, can he still use contact lens?
Should not be related to contact lens. I think they can, yes.
They can.
So here is the interesting thing, right? Usually the therapy for cataract is they're going to remove the existing lens, an artificial lens in there. And the artificial lens is only going to have a single focal point. It is not going to be bionic eye, or it's not going to have accommodation in it. So there are accommodating lenses available, but that's not the industry standard. So if you use an accommodating contact lens on top of that, it's possible, or you can use a bifocal glasses after cataract.
Right. Okay. Thank you.
Dr. Harish, we could end the meeting session.
Yes, sir.
Dr. Harish.
I don't find any other questions here.
It was a very amazing and a very great lecture by Dr. Srinivas Anasoge, live from San Francisco. He has taken a lot of trouble in preparing for this presentation from quite a few times. He's on board as an advisory committee member, international advisory committee member on our international conference, ICACM 2020. I've been working in contact with him to make this plenary session possible. So I think today he is able to present all his work that he has done in the past at Rutgers and Georgia Tech, as well as now presently doing it with Google Life Sciences. So on behalf of the organizing committee and behalf of the Darwin Psychology Meeting, it's a privilege and joy to thank our proud alumni for giving this excellent plenary session on microfluidics, MEMS, and microfabrications. Thank you, Dr. Srinivas, for that wonderful lecture, and plenary session was really interesting. And thank you. I thank all the participants here, Dr. CPS Prakash, Principal, Dr. Harish, and all other head of the departments, the organizing committee members for making this plenary session possible. It is for the students here and the participants of the conference here. This is hopefully been very benefited, in terms of the takeaways are really great in terms of the new technology on microfabrications. And I thank you, Dr. Srinivas.
Thank you, Dr.
Thank you very much.
All the best.
Thank you, sir. Thank you very much.
All the best. All the best. Thank you. Thank you. Shall we end the meeting, Dr. Harish?
Yes, sir. Thank you. So thank you all the participants. Now we have come to the end of the session. Now you can join the individual sessions using the links provided in the WhatsApp group. So you can now log out. Thank you.
Thank you. Thank you. Thank you all.